What is the role of polygenic risk scores (PRS) in predicting disease risk, and how do they integrate genetic data across multiple loci? Explain the key assumptions of PRS and the potential limitations in clinical application. Answer: polygenic risk scores (PRS) aggregate genetic information from multiple loci to estimate an individual's genetic predisposition to a specific disease. Each locus contributes a small effect, and the combined effects of these loci are thought to sum up to influence complex traits or diseases. The PRS is typically calculated by summing the risk alleles at various loci, weighted by the effect size of each allele derived from genome-wide association studies (GWAS). Key assumptions of PRS include additive model. The effects of individual loci are assumed to add up linearly. Stable effect sizes. The effect sizes of SNPS, single nucleotide polymorphisms, are assumed to be consistent across different populations. Limitations in clinical application include population stratification. Differences in allele frequencies across populations can lead to inaccurate risk predictions if the PRS model is not adjusted for these variations. Missing heritability. PRS may not account for all genetic variants, influencing a trait leading to lower accuracy in some cases. Mr. Rosenberg, a member of the Ashkenazi Jewish population, is concerned about his genetic risks for diseases prevalent in his ethnic group. What is the concept of founder effects, and how does it influence the prevalence of certain genetic disorders within isolated populations like the Ashkenazi Jews? Answer: the founder effect refers to the reduced genetic diversity that occurs when a small group of individuals from a larger population establishes a new population. This new population may have an overrepresentation of certain genetic variants, leading to an increased frequency of specific diseases or traits within the group. In the case of Ashkenazi Jews, many genetic disorders are more prevalent due to historical bottlenecks and small founding populations. These populations carried specific mutations that became common within the group over generations. Examples of genetic disorders more common in Ashkenazi Jews include Tay-Sachs disease, cystic fibrosis, and Canavan disease. The founder effect means that individuals from isolated populations may carry a higher risk for specific genetic disorders due to the concentration of mutations in the founding group. Genetic screening is important in these populations to identify carriers and manage risk for offspring. Explain the role of hedgehog signaling in development and describe how mutations in this pathway can result in developmental disorders. Answer: the hedgehog HH signaling pathway is crucial in regulating embryonic development, cell differentiation, tissue patterning, and organogenesis. It plays a significant role in the development of structures such as limbs, the neural tube, and the brain. In the normal pathway, the hedgehog protein binds to a receptor called patched, which then releases inhibition of another receptor, smoothen. This allows downstream signaling that activates transcription factors involved in the expression of target genes. Mutations in the hedgehog pathway can disrupt normal development and lead to various congenital disorders such as holoprosencephaly, failure of the forebrain to divide into two hemispheres, often caused by mutations in the SHH gene, which encodes the hedgehog ligand. Basal cell carcinoma (BCC). Normal activation of the pathway can lead to unregulated cell growth, contributing to cancer. These disorders demonstrate the critical role of tightly regulated hedgehog signaling in maintaining normal development. How does the neutral theory of molecular evolution differ from the traditional Darwinian view of natural selection, and what implications does it have for understanding genetic variation in populations? Answer: the neutral theory of molecular evolution proposed by Motoo Kimura suggests that much of the genetic variation observed in populations is due to random genetic drift rather than natural selection. According to this theory, the majority of mutations are neutral, meaning they do not affect an organism's fitness and are not subject to selective pressure. In contrast, traditional Darwinian evolution emphasizes that natural selection is the primary force driving adaptive changes in a population, with advantageous mutations increasing in frequency due to better survival or reproductive success. The implications of the neutral theory for genetic variation are significant. Genetic drift. Neutral mutations accumulate in a population over time through genetic drift, particularly in small populations, leading to genetic diversity that is not necessarily adaptive. A molecular clock. The rate of accumulation of neutral mutations can be used to estimate the time of divergence between species, forming the basis for the molecular clock hypothesis. Overall, the neutral theory underscores that genetic variation can exist without providing any selective advantage or disadvantage. Dr. Lee is conducting a study on the genetic diversity of a remote island population. He notices that the population exhibits significant inbreeding, and he is concerned about how this may affect the study's results. What statistical tools could Dr. Lee use to analyze the genetic data and assess the degree of population stratification and inbreeding? What are the potential implications for his findings? Answer: Dr. Lee can use the following statistical tools to assess inbreeding and population stratification. Plink. This tool can be used to analyze genetic data and assess relatedness between individuals using identity by descent calculations, which helps detect inbreeding. Statistics, such as FIS, the inbreeding coefficient, measure the degree of inbreeding in a population. A higher FIS value indicates more inbreeding. Dr. Lee could use these values to estimate the inbreeding coefficient of the population. Principal component analysis (PCA). PCA can help visualize population structure and stratification by identifying clusters of individuals based on genetic similarity, which is essential for identifying subpopulations. The implications of finding significant inbreeding in the population include the potential for reduced genetic diversity, which could impact the study's conclusions. Inbreeding can lead to inbreeding depression, where harmful recessive traits are more likely to manifest, leading to reduced fitness and health problems. How does RNA sequencing (RNA-seq) enhance our understanding of gene expression during development, and what advantages does it offer over traditional gene expression analysis techniques? Answer: RNA sequencing (RNA-seq) allows for the comprehensive analysis of gene expression by sequencing all the RNA molecules present in a sample. It is particularly useful in developmental genetics for understanding the temporal and spatial regulation of gene expression during various stages of development. RNA-seq provides several advantages over traditional methods ICOIA or microarrays, transcriptome-wide analysis. Unlike microarrays, which can only detect known genes, RNA-seq captures the expression levels of all genes, including novel or previously unannotated ones. Higher sensitivity RNA-seq can detect low abundance transcripts that might be missed by traditional methods. Quantification of isoforms. It enables the identification of alternative splicing events, which is essential for understanding how different isoforms contribute to developmental processes. Overall, RNA-seq has revolutionized developmental genetics by allowing researchers to uncover the complex regulatory networks that control gene expression in development. Explain the significance of single-cell RNA sequencing in developmental biology, and how does it overcome the limitations of bulk RNA sequencing? Answer: Single-cell RNA sequencing (RNA-seq) is a powerful technique that allows the examination of gene expression at the level of individual cells. This is particularly significant in developmental biology, where cellular diversity and heterogeneous gene expression are key to understanding processes like differentiation and tissue formation. In traditional bulk RNA sequencing, the RNA from a population of cells is pooled together, making it difficult to distinguish the gene expression profiles of different cell types or stages of differentiation. In contrast, RNA-seq captures the transcriptomic profiles of individual cells, revealing heterogeneity within cell populations and identifying rare or transient cell types that may be crucial during development. Key benefits of RNA-seq include identifying novel cell types. It can uncover previously unknown cell types or states within tissues, tracking developmental trajectories. It allows researchers to map how cells transition between different states during development. In summary, RNA-seq provides a resolution that is unmatched by bulk RNA-seq, offering deeper insights into developmental processes. What are the primary challenges in implementing population-specific screening programs, such as for Tay-Sachs disease, and how do these challenges impact the effectiveness of the screening? Answer: Population-specific screening programs aim to identify carriers of genetic diseases more prevalent in certain ethnic groups, such as Tay-Sachs disease in Ashkenazi Jews. However, several challenges affect their implementation. Ethical considerations. Screening for genetic conditions raises ethical issues such as the right to know one's genetic status and the potential for discrimination or stigmatization based on genetic information. Cost and accessibility. Screening programs can be expensive, and ensuring that the population has access to genetic counseling and follow-up care is critical to the program's success. Carrier frequency. For diseases like Tay-Sachs, it's crucial to know the carrier frequency in the population to ensure that the screening is both cost-effective and efficient. Screening may be less effective in populations with low carrier frequencies. Pre- and post-test counseling. Proper counseling is essential to help individuals understand their genetic risk, the meaning of their results, and the implications for family planning. Explain the concept of the molecular clock and how it can be used to estimate evolutionary divergence times between species. What assumptions must be made for this approach to be valid? Answer: the molecular clock is a technique used to estimate the time of divergence between species by analyzing the accumulation of genetic mutations over time. The underlying assumption is that genetic mutations accumulate at a relatively constant rate, allowing the molecular differences between species to serve as a clock to estimate when two species diverged from a common ancestor. For the molecular clock to be valid, several assumptions must be made. Constant mutation rate. Mutations accumulate at a constant rate over time across different lineages. Neutral mutations. The mutations used in the molecular clock must be largely neutral, meaning they do not significantly affect fitness and are not subject to natural selection. Calibration points. Fossil evidence or known divergence times must be used to calibrate the molecular clock, ensuring that the estimated divergence times are accurate. The molecular clock is particularly useful in situations where fossil records are sparse, and it can provide insights into the timing of evolutionary events. How does the study of genetic disorders in the Amish population exemplify the founder effect, and what are the potential implications for genetic counseling in such communities? Answer: the Amish population, due to its historical isolation and limited genetic diversity, exhibits several genetic disorders at higher frequencies than the general population. The founder effect is evident in this community as certain genetic mutations were passed down from a small group of founding ancestors, resulting in a higher prevalence of specific disorders. Examples of disorders more common in the Amish population include mucopolysaccharidosis, maple syrup urine disease, and Ehlers-Danlos-Creveld syndrome. The implications for genetic counseling in such communities include carrier screening. Genetic counseling becomes crucial for identifying carriers of recessive diseases and informing reproductive decisions. Reduced genetic diversity. Long-term inbreeding may increase the expression of recessive genetic disorders, necessitating careful monitoring and counseling. Cultural sensitivity. Genetic counseling must be tailored to respect the Amish community's cultural values, including their preference for maintaining privacy and avoiding intervention in genetic matters. A team of researchers plans to use CRISPR-Cas technology to treat cystic fibrosis by targeting the CFTR gene. However, they encounter a challenge with off-target effects. Which strategies can the researchers use to minimize off-target effects, and how do these strategies align with regulatory guidelines? Answer: minimizing off-target effects in CRISPR-Cas applications involves several strategies. GuideRNA gRNA design use computational tools to design highly specific gRNA minimal sequence similarity to other genomic regions. The FDA emphasizes rigorous in silico analysis to predict and avoid unintended cleavage sites. Modified Cas proteins employ high-fidelity variants of Cas, E, G, Espers hypochas that are engineered for increased specificity. These modified enzymes meet regulatory requirements for minimizing genotoxicity. Delivery methods use localized delivery mechanisms to target only affected tissues. For instance, lipid nanoparticles or tissue-specific promoters can confine CRISPR activity. Validation techniques perform whole genome sequenceability at GuideSeq to confirm the absence of off-target edits post-treatment, a practice encouraged by the FDA under the 21st Century Cures Act. Which factors should be considered when selecting a viral vector for gene therapy, and how do these factors influence clinical trial outcomes? Answer: key factors in viral vector selection include tropism. Choose a vector that specifically targets the intended tissue. For example, adeno-associated viruses (OVs) are ideal for muscle and liver targeting. Immunogenicity. Assess and minimize immune responses to avoid rejection. Lentiviruses tend to have lower immunogenicity compared to adenoviruses. Integration versus episomal maintenance. Lentiviral vectors integrate into the host genome, providing stable expression, whereas AA versus remain episomal, reducing the risk of insertional mutagenesis. Payload capacity. Match the vector's capacity with the therapeutic gene size. AA versus are suitable for small genes, 4.7 kilobytes, while lentiviruses can carry larger genes. Regulatory compliance. Ensure adherence to 21 CFR Part 312 for investigational new drug applications, focusing on vector safety and manufacturing quality. Selecting an appropriate vector affects transduction efficiency, therapeutic outcomes, and patient safety, all of which are critical in clinical trial design and success. Dr. Susan Harper is designing a phase II clinical trial to evaluate a new gene therapy for spinal muscular atrophy, SMA. During preliminary review, she identifies a patient population with varying SMA gene copy numbers and a history of previous OV-based treatments. What steps should Dr. Harper take to optimize patient selection and ensure compliance with FDA guidelines for clinical trials? Answer: Dr. Harper should address the following: stratify patients by genotype, use genetic testing to classify patients based on SMADine copy number. Patients with 1, 2 copies are likely more responsive to the therapy. The FDA encourages targeted patient selection to improve efficacy evaluations, account for pre-existing immunity, screen for neutralizing antibodies against AAV vectors as prior exposure may compromise vector delivery. Patients with high antibody titers should be excluded or considered for alternative vector systems. Implement biomarker analysis. Utilize SMM protein levels as a biomarker to assess baseline conditions and monitor therapeutic response. Ethical and regulatory considerations. Adhere to ICHR good clinical practice guidelines to ensure ethical patient inclusion and rigorous trial oversight. Dr. Harper's approach should balance scientific rigor with patient safety, ensuring that trial outcomes are both meaningful and compliant with FDA standards. How do specific HLA alleles contribute to the pathogenesis of autoimmune diseases, and why is HLA typing crucial in personalized medicine? Answer: human leukocyte antigen HLA alleles play a central role in autoimmune diseases by presenting antigens to T cells. Association with autoimmune disease. A-TSHR is strongly linked to ankylosing spondylitis, while A-TSHR increases the risk for rheumatoid arthritis. These alleles skew immune responses, leading to autoimmunity. Molecular mechanisms. Certain HLA alleles preferentially bind self-peptides, leading to aberrant activation of autoreactive T cells. Cross-reactivity with pathogen-derived peptides. Molecular mimicry can also trigger autoimmunity. Personalized medicine. HLA typing allows for targeted therapies, such as excluding patients with A-TSHR-5701 from abacavir treatment due to hypersensitivity risks. The integration of HLA data into treatment plans aligns with FDA guidelines under the personalized medicine framework. A pharmaceutical company is testing a new CAR-T cell therapy for relapsed acute lymphoblastic leukemia. One patient developed cytokine release syndrome (CRS) post-infusion. What immediate steps should the medical team take to manage CRS, and how do these align with clinical guidelines? Answer: management of CRS includes grading the severity. Use the ASTCT consensus grading system to classify CRS severity based on fever, hypotension, and hypoxia. Administering tocilizumab for grade 2 or higher CRS. Administer tocilizumab, an IL-6 receptor antagonist, as recommended by the FDA-approved CAR-T cell therapy guidelines. Supportive care. Provide intravenous fluids for hypotension and oxygen therapy for hypoxia. Monitor and adjust treatment regularly. Monitor inflammatory markers e.g. CRP, ferritin to guide further interventions. This approach ensures patient safety while adhering to FDA recommendations for post-infusion monitoring in cellular therapies. Gene therapy has the potential to revolutionize the treatment of genetic disorders, but regulatory bodies face significant challenges in balancing innovation with patient safety. What are the primary regulatory challenges faced by gene therapy products, and how do the FDA and EMA address these concerns? Answer: gene therapy faces regulatory challenges in several areas. Safety and long-term monitoring. Regulatory bodies demand rigorous preclinical and clinical safety data, including potential long-term risks such as insertional mutagenesis, off-target effects, and immune responses. Both the FDA and the European Medicines Agency (EMA) require ongoing monitoring of gene therapy patients even after market approval under the FDOS-RE/MS Risk Evaluation and Mitigation Strategy Program. Manufacturing and quality control. The complexity of gene therapy products, including viral vector production and gene editing, demands adherence to good manufacturing practice (GMP). Both regulatory bodies emphasize transparency in the manufacturing process, with the FDA issuing guidance under 21 CFR Part 211 to ensure product consistency and purity. Clinical trial design. The lack of standard protocols for gene therapies complicates trial design, as each therapy may require unique endpoints and patient populations. The FDOS 21st Century Cures Act provides flexibility in clinical trial design, allowing for more adaptive approaches while maintaining safety standards. Ethical considerations and public perception. The FDA and EMA address ethical concerns related to gene editing, particularly germline editing, by enforcing strict guidelines under the Gene Editing and Innovation Act and GMO (Genetically Modified Organisms) regulations. Balancing innovation with patient safety requires extensive collaboration between the scientific community, regulatory bodies, and ethical committees. How does CRISPR-Cas technology specifically address challenges in treating polygenic diseases, and what regulatory and ethical concerns must be considered? Answer: CRISPR-Cas offers a precise genome editing tool that holds promise for treating polygenic diseases. However, addressing challenges and regulatory concerns requires careful consideration. Polygenic diseases and CRISPR polygenic diseases involve multiple genes and complex interactions, making them difficult to treat with traditional gene therapy. CRISPR can target multiple loci simultaneously, but the complexity of polygenic regulation poses challenges in ensuring therapeutic precision. Precision and off-target effects. CRISPR's ability to edit multiple genes introduces the risk of off-target effects, which may lead to unintended mutations in other parts of the genome. Regulatory bodies require rigorous testing for off-target mutations as outlined in FDA's genetic modification guidelines. Ethical concerns. The use of CRISPR in humans, especially for polygenic diseases, raises significant ethical questions regarding consent, germline editing, and unintended long-term consequences. The International Summit on Human Gene Editing has called for a moratorium on heritable genome editing until further ethical and safety concerns are addressed. Regulatory oversight. The FDA's oversight includes guidelines under the Gene Therapy Clinical Trial Guidance, ensuring clinical trials rigorously evaluate the safety and efficacy of CRISPR-based therapies before approval. These challenges underscore the importance of ethical considerations and stringent regulatory frameworks when applying CRISPR in gene therapy. What are the genetic mechanisms behind severe combined immunodeficiency (SID) and DiGeorge syndrome, and how do these mechanisms influence treatment strategies? Answer: SCID and DiGeorge syndrome are both primary immunodeficiency disorders with distinct genetic causes and treatment strategies. SCID mechanism. SCID is a group of disorders characterized by the absence of functional T cells, leading to a severe lack of immune response. It is often caused by mutations in genes such as ILG-X-linked SID-ADA adenosine deaminase deficiency or RAGWO recombination activating genes. Treatment involves hematopoietic stem cell transplantation (HCT) or gene therapy. The FDA-approved Strimveli's gene therapy for ADA-SID is an example of how gene therapy has improved patient outcomes. DiGeorge syndrome mechanism. DiGeorge syndrome results from a deletion on chromosome 20.2, leading to thymic hypoplasia and T-cell deficiency. It also affects other organ systems, including the heart and parathyroid glands. The treatment involves a combination of thymic transplant and immunoglobulin therapy to correct immune function. Gene therapy is also being explored to address the underlying genetic defect. Impact on treatment strategies. In both conditions, genetic testing plays a critical role in diagnosis and treatment planning, as it informs the clinician about the specific mutation and its implications for therapy. Gene therapy trials are being conducted for both diseases, with regulatory approval based on safety and efficacy data under the FDA's orphan drug designation for rare diseases like SCID. By identifying the underlying genetic cause, these treatments aim to restore normal immune function and provide long-term solutions to immunodeficient patients. What are the key immunogenetic considerations when using CAR-T cell therapy for cancer treatment, and how do these considerations guide clinical decisions? Answer: CAR T-cell therapy has emerged as a promising treatment for various cancers, particularly hematologic malignancies. The following immunogenetic considerations are critical. Tumor antigen selection. CAR T-cells are engineered to recognize specific antigens on cancer cells, such as CD, in B-cell malignancies. Careful selection of tumor-specific antigens is crucial to avoid targeting normal cells, which could result in autoimmunity. T-cell receptor modification. The genetic modification of T cells to express a chimeric antigen receptor (CAR) is a key element of CAR T therapy. The TCR signaling pathway must be properly calibrated to enhance the anti-tumor response while minimizing toxicity. Cytokine release syndrome (CRS). CRS is a major side effect of CAR T cell therapy. The immune response generated by CAR T cells can cause an overproduction of pro-inflammatory cytokines, leading to fever, hypotension, and organ dysfunction. Managing CRS with cytokine inhibitors like tocilizumab is essential. Genetic testing for tumor mutations. Comprehensive genomic profiling of the tumor is critical to identify mutations and potential resistance mechanisms. This helps to tailor the CAR T therapy to the patient's specific cancer type and genetic profile. FDA approval and guidelines. CAR T therapies must undergo rigorous testing as per FDA's guidance on gene therapy. The therapy must meet safety standards related to T-cell expansion, cytokine release, and off-target effects. By addressing these immunogenetic factors, clinicians can maximize the efficacy of CAR T cell therapy while minimizing adverse effects. How do host pathogen genetic interactions influence the outcome of infections, and what implications do these interactions have for developing precision medicine strategies for infectious diseases? Answer: host pathogen genetic interactions play a significant role in the outcome of infections. Host genetic factors variations in the HLA and immune system genes influence how individuals respond to infections. For example, the CCRD mutation provides resistance to HIV infection by preventing the virus from entering cells. Polymorphisms in immune genes such as IL, TNF, and IL can affect inflammation and the immune response to pathogens like Mycobacterium tuberculosis. Pathogen genetic factors: pathogens themselves undergo genetic changes to evade the immune system. For example, influenza virus and HIV frequently mutate their surface proteins to avoid recognition by the host immune system. Precision medicine implications: understanding these genetic interactions allows for genetic profiling of patients to predict susceptibility and response to specific infections. In the case of antimicrobial resistance (AMR), genetic information from both the host and pathogen can guide the choice of antibiotics or antivirals, reducing the risk of ineffective treatment. Regulatory approaches: the FDA's Precision Medicine Initiative encourages the integration of genetic data into patient care, allowing for more effective treatments and the development of personalized vaccines. These interactions are pivotal in advancing personalized medicine strategies for infectious diseases, improving both treatment outcomes and public health. Explain the 5-tier classification system recommended by GAM for variant classification in genetic testing. Discuss the impact of each classification on clinical decision-making. Answer: the GAM guidelines classify genetic variants into 5 categories to aid clinicians in interpreting results and guiding patient management. Pathogenic (P). These variants are definitively associated with disease. Clinical interventions such as targeted therapy, genetic counseling, or family screening are often necessary. Likely pathogenic (LP). Variants with substantial evidence supporting their pathogenicity, but not definitive. Clinicians may take clinical action based on clinical presentation, family history, or other supporting evidence. Uncertain significance. Thus, these variants lack sufficient evidence to determine their clinical significance. While further research or testing may resolve the classification, VUS should be clearly documented and the patient should be counseled on the uncertainty. Likely benign (LB). Variants that are unlikely to be associated with disease but without conclusive evidence. Clinically, these variants usually do not warrant action, though some may still require monitoring depending on family history. Benign (B). These variants have been proven not to cause disease. No clinical action is necessary, and these variants are often not reported unless requested. Impact on clinical decision-making. Classifying variants helps clinicians prioritize which findings to act upon. Pathogenic and likely pathogenic variants typically lead to immediate clinical actions such as patient management, genetic counseling, or additional screening for family members. Variants classified as VUS require careful consideration and often further testing, while benign or likely benign variants typically do not require follow-up, reducing unnecessary patient anxiety or medical intervention. Accurate classification is critical for patient care and reduces the risk of unnecessary treatment or misdiagnosis. Describe the primary sources of errors in the pre-analytical, analytical, and post-analytical phases of genetic testing. Propose strategies to mitigate these errors. Answer: errors in genetic testing can arise in the following phases. Pre-analytical errors. These occur before the actual testing begins and include errors like incorrect sample labeling, sample contamination, improper storage, or insufficient collection techniques. Mitigation. Implement strict sample labeling protocols with automated tracking and ensure proper storage conditions, e.g. temperature control. Training for healthcare providers on sample collection and handling is also essential to prevent contamination. Analytical errors. These errors occur during the testing process, such as issues with PCR amplification, sequencing errors, or machine malfunction. Mitigation. Regular equipment calibration, inclusion of control samples in each assay, and use of validated testing platforms reduce these risks. Automated systems for data analysis can also minimize human error in interpreting results. Post-analytical errors. These errors arise during result interpretation, such as incorrect conclusions drawn from sequencing data, data transcription errors, or misclassification of variants. Mitigation. Implement double-checking procedures for reporting, peer-reviewed systems for test results, and use of bioinformatics tools that support variant interpretation. Documentation should be meticulous, and staff should receive continuous training to keep up with evolving standards. Proficiency testing and quality assurance programs are essential in all stages to monitor and correct errors in testing processes. A couple seeks pre-implantation genetic testing (PGT) to select embryos without a gene associated with a predisposition to cancer. However, they also request selection for non-disease traits such as eye color and height. Discuss the ethical considerations of this request and recommend how the clinician should proceed. Answer: ethical considerations. Autonomy. The couple has the right to make reproductive decisions based on their values, but this must be weighed against the potential societal impacts of their choices. Beneficence and non-maleficence. While selecting for disease-free embryos is in line with promoting the well-being of the child, selecting for non-medical traits like eye color may cross ethical boundaries. This could contribute to design at babies and societal pressure to conform to certain physical standards, raising concerns about equity and fairness. Justice. Such choices might exacerbate social inequalities by privileging certain genetic traits and further stratifying access to advanced reproductive technologies. Recommendation. The clinician should emphasize the ethical implications of non-medical trait selection and guide the couple toward focusing on health-related genetic information. While autonomy is paramount, the clinicians should inform the couple of the broader ethical debate surrounding such requests and ensure that all decisions are made with full understanding of the potential long-term consequences. Wise participation in external quality assurance (ECHA) programs critical for laboratories performing genetic testing. Identify 3 benefits of EQA participation. Answer: external quality assurance (ECHA) programs are essential for ensuring that laboratories maintain the accuracy, consistency, and reliability of genetic testing. Benefits of EQA participation. Benchmarking performance. EQA programs allow laboratories to compare their results with other laboratories performing the same tests. This helps identify any discrepancies in the testing process and improve laboratory practices. Regulatory compliance. Participation in EQA is often required by regulatory agencies like the Clinical Laboratory Improvement Amendments (CLIA) in the United States. This helps laboratories maintain accreditation and ensures compliance with federal standards for accuracy and reliability. Quality improvement. The feedback received from EQA programs serves as a tool for continuous quality improvement. Laboratories can use the feedback to refine protocols, correct errors, and enhance staff training, ultimately improving the quality of patient care. By participating in EQA, laboratories also ensure that their test results are credible and trusted by clinicians and patients, further reinforcing the integrity of the diagnostic process. Compare and contrast the roles of the FDA and EMA in regulating genetic testing. Highlight key differences in their approach. Answer: FDA US. The FDA is responsible for ensuring that genetic tests meet rigorous standards for safety, efficacy, and quality before they are marketed in the United States. This includes pre-market approval for genetic tests that are considered high risk, particularly those used for medical diagnostics or treatment decisions. The FDA evaluates test performance, clinical validity, and potential harm before a test can be approved for use in clinical settings. EMA Europe. The European Medicines Agency (EMA) is responsible for ensuring that genetic tests marketed in the European Union meet safety and efficacy standards. However, the regulatory environment in Europe allows more flexibility than the FDA in certain areas, especially for low-risk genetic tests. The In Vitro Diagnostic Devices Regulation, after which the EMA oversees, establishes stricter rules for tests categorized as high risk. Key differences. The FDA has a more centralized, top-down approach with tighter control over genetic test approvals, especially for diagnostics. The EMA operates within a more decentralized system with different member states involved in the regulatory process, allowing some tests to be marketed with less stringent oversight in certain cases. Pre-market approvals are mandatory for high-risk tests in the US, but more flexible in the EU, depending on risk categories. What are the primary goals of genetic counseling in a clinical genetic setting? Answer: the primary goals of genetic counseling are to help individuals and families understand and make informed decisions about genetic testing, inheritance patterns, and the implications of genetic conditions. Specific goals include risk assessment. Counselors help assess the genetic risk of inherited conditions based on family history and genetic testing. Information delivery. Clear, understandable information about genetic conditions, including possible outcomes, testing options, and treatment possibilities. Decision-making support. Genetic counselors provide support in decision-making, guiding patients through options like genetic testing, reproductive choices, or preventive health measures. Psychological support. Counselors offer emotional support, helping patients cope with the implications of genetic testing, such as learning about a diagnosis or dealing with uncertainty. Counseling is patient-centered and aims to empower individuals by offering the necessary resources to make informed choices about their health and the health of their family members. Explain the importance of creating a family pedigree in genetic counseling. How does pedigree information contribute to clinical decision-making? Answer: a family pedigree is a visual representation of a family history that displays relationships between family members and the inheritance of traits or genetic conditions. It is crucial in genetic counseling for several reasons: identifying inheritance patterns. Pedigrees help genetic counselors and healthcare providers identify whether a condition follows autosomal dominant, autosomal recessive, X-linked, or other inheritance patterns, which informs the likelihood of passing the condition to offspring. Risk assessment. By analyzing family structures and the occurrence of disease in relatives, counselors can estimate an individual's risk of developing or inheriting genetic disorders. Guiding testing and management. Pedigrees help determine whether genetic testing is warranted and which tests might be most appropriate based on the familial pattern of disease. Creating a detailed family pedigree contributes to a personalized genetic evaluation, ensuring that medical decisions are based on comprehensive family health data. What role does pharmacogenomics play in personalized medicine? Provide an example of how pharmacogenomic data can inform drug therapy. Answer: pharmacogenomics is the study of how an individual's genetic makeup influences their response to drugs. It plays a central role in personalized medicine by enabling the selection of drugs that are more likely to be effective and have fewer side effects based on a person's genetic profile. Example: one of the most well-known applications of pharmacogenomics is in the use of the drug warfarin, an anticoagulant. Variants in the SIC and VCOR genes can affect how an individual metabolizes warfarin, influencing their risk of bleeding or clotting. By testing for these variants, clinicians can tailor the warfarin dosage to the patient's genetic profile, minimizing adverse reactions and improving treatment outcomes. Pharmacogenomics thus represents a major shift in medicine, moving away from the one-size-fits-all approach to a more individualized, targeted treatment strategy. Differentiate between missense and nonsense mutations. How do these mutations affect protein function and contribute to disease? Answer: missense mutation. A missense mutation occurs when a single nucleotide change leads to the incorporation of a different amino acid into the protein. This can alter the protein's structure and function, sometimes rendering it non-functional or causing it to function abnormally. For example, in sickle cell anemia, a missense mutation in the HBB gene leads to the substitution of valine for glutamic acid, resulting in abnormal hemoglobin molecules that cause red blood cells to sickle. Nonsense mutation. A nonsense mutation occurs when a nucleotide change creates a premature stop codon, truncating the protein. This often results in a non-functional protein or one with significantly reduced function. For example, in cystic fibrosis, a nonsense mutation in the CFTR gene may lead to a shortened, non-functional protein that disrupts chloride ion transport across cell membranes. Both types of mutations can lead to serious genetic disorders, though the impact on protein function and clinical severity depends on the specific mutation and the affected gene. What are the primary ethical considerations in genetic testing, particularly in the context of testing minors? Answer: genetic testing, especially when involving minors, raises several ethical issues, including informed consent. Informed consent is a fundamental ethical requirement in genetic testing. For minors, obtaining consent from parents or guardians is necessary, but the child's assent agreement should also be considered when appropriate, depending on their age and understanding. Autonomy and privacy. Testing minors may involve issues of autonomy, as children are not fully capable of making independent healthcare decisions. Protecting the child's privacy and ensuring that genetic information is only shared with authorized individuals are crucial ethical considerations. Potential psychological impact. The knowledge of a genetic predisposition to certain diseases may be difficult for minors to comprehend, and it could lead to anxiety, stress, or stigmatization. Counselors must carefully assess whether testing is in the best interest of the child and ensure that the results are communicated in a way that minimizes emotional harm. Beneficence versus non-maleficence. Healthcare providers must weigh the potential benefits of genetic testing against the risks. For example, testing for a genetic disorder may enable early intervention but could also cause harm if it leads to discrimination or unnecessary treatments. Genetic discrimination. There is a risk that the results of genetic tests may lead to discrimination in employment, insurance, or social situations. The Genetic Information Non-Discrimination Act, (GINA) in the US, and similar laws in other countries aim to protect individuals from such discrimination. Ethical decisions must balance the potential benefits of genetic knowledge with the rights and well-being of the minor and the family.